You may have always wondered why studying can actually make your memory of a specific subject better. And the best way to explain this is by looking at the processes that take place in the brain. The brain is an organ that is found in the human body, and it has several structures inside of it that have many functions. So one of the structures is the hippocampus, and I will signal it out right now. So the hippocampus is a specific structure in the brain that relates to memory formation and retrieval. It is also the structure where long-term potentiation was discovered, and it was discovered in 1973. So that is the process that I'll be talking about in this presentation. So inside the brain, there are various types of cells. One type of cell found in the brain is the neuron, and it's very important when it comes to long-term potentiation, because this is where it takes place. So basically, for an action potential to travel down a neuron, it has to go from the dendrites to the cell body, to the axon tips, and then it will turn from an electrical signal to a chemical signal in the synapse, which is this red circle that you can see right there, and will then transmit the message as another action potential to the next neuron. So it will go electrical, chemical, and then electrical again. And where this chemical process is taking place is where neurotransmitters are released. So you can see the little blue dots in the picture. Those represent the neurotransmitters that are being released to the next neuron. The first neuron in this picture would be called the presynaptic neuron, which comes before the synapse. And the second one is the postsynaptic neuron, which comes after the synapse. So that is important because for a message to be passed to the next neuron and through the next neuron, it will have to increase the threshold that the neuron has, which at resting potential will be around -70 millivolts. For depolarization to take place, which is what I'm talking about, it will have to be around -55 millivolts. So it will become more positive and will become less polar. And this process only takes place when ion channels open, because ions are found around the neurons, and the influx of ions into the neuron will make it more positive. So here you can see a diagram of the actual process that will take place. The neuron that is found at the top will be the presynaptic neuron, and the neuron that is found in the bottom will be the postsynaptic neuron. Here you can also see a key of the different ions that there are, or well, the ones that are important for this. And the blue dots inside the vesicles that are found in the presynaptic neuron are glutamate. So that's a neurotransmitter. In this part, what I'm showing is what happens when a low frequency stimulus goes through the presynaptic neuron. So glutamate will be released into the fluid that is outside the neuron, and it will connect to the AMPA receptor, which is the red one, and also to the NMDA receptor, which is the blue one. The AMPA receptor will let in sodium ions, which will make the postsynaptic neuron slightly more positive, but not depolarized. And even though the NMDA receptor is activated, it does have a magnesium ion that is blocking the calcium ions from flooding in. So in this second example, long-term potentiation will take place because the higher frequency stimulus will go through the presynaptic neuron, which will then make more glutamate be released. So here you're gonna see how more glutamate is being released into the space between the neurons, so into the synapse. And again, they're gonna bind to the AMPA receptor and to the NMDA receptor. So that activates both of them. Now, when the AMPA receptor starts having sodium ions flood in, that's gonna make the membrane of the postsynaptic neuron very depolarized, because there's gonna be a lot of positive ions inside of it and not a lot outside. So when this starts happening, the magnesium ion that is blocking the NMDA receptor is going to be repelled, and then calcium ions will be able to go into the cell. And calcium ions are known as secondary messengers. So not only are they going to make the inside of the cell more positive, but they're going to bind to proteins inside of the cell, and they're going to make more AMPA receptors be available for future depolarization, which means that in the next times that the cell is going to be depolarized, there's going to be more AMPA receptors letting sodium in, and that means that it's going to be easier to depolarize the cell. This process that I just spoke about is the first process that takes place in long-term potentiation. And the second phase is called the late phase. So the first one is early phase and the second is late phase. And late phase happens when more calcium ions go into the cell. So these calcium ions will bind to more proteins. And not only are AMPA receptors going to be produced, but on top of that, growth factors are going to be produced. And more synapses are going to be formed between these two neurons, which means that in the end, the connections between them are going to be way stronger. So that is what happens when you're learning. And this is how you become smarter and actually become able to memorize certain types of knowledge if you rehearse it more. Now I'm going to be showing you an animated version of the process I just explained, and this is from a website called The Brain from Top to Bottom. So here you see how there's an option to see transmission with a unique action potential. So that's when it's low frequency and it's not gonna cause long-term potentiation. So the glutamate is binding to the AMPA and the NMDA receptors, and sodium did go through the AMPA receptor, but the magnesium ion did not get repelled from the NMDA receptor because there's not enough sodium ions inside of the cell to repel it. So when you try again with the other option, which is the high frequency stimulation producing a long-term potentiation, what happens is that this is going to release way more glutamate than was released initially. So, that's what's taking place right now. And the sodium ions are going to go into the AMPA receptor, which will repel the magnesium ion from the NMDA channel, and the calcium ions will flow in, which will then bind to the proteins and create more AMPA receptors. That will only be early phase, and then late phase will also create more AMPA receptors, but it will also make growth factors that will make more synapses between the two neurons and make the connection stronger. Thank you for listening to my presentation.